Okay, so welcome everyone to the July webinar of PAASTA, so the Paleoproteomics and Archaeological Society for Techniques and Advances. So the purpose of these webinars is to inspire connection and communication between members of our community. So please feel free to ask the speaker questions at the end of the talk. However, these webinars may contain some published or unpublished data, and resulting kind of conversations and content should not be shared without the express permissions of the speaker. So although these webinars are informal, the speaker should be contacted directly if you're seeking interviews or statements for reporting. So finally, we want to encourage a safe and positive space for the sharing of knowledge and information in our community. So we ask everyone to abide by our code of conduct, which I'll post a link to in the chat. And so this week, we're very happy to welcome Evan Saitta, who's joining us from the University of Chicago. And he's going to be presenting his paper, which was published in eLife. And I'm going to post a link to that in the chat as well. So I'm going to hand over to Evan, who's going to present Cretaceous dinosaur bone contains recent organic material and provides an environment conducive to microbial communities. So Evan, if you want to share your screen.
All right, let me go ahead. Oh. Can everyone see?
Yeah, perfect.
All right. Well, I put the title slide as the different paper, the more recent paper on eggshell, but that's fine because I'm going to be talking about both studies anyway, so it doesn't really matter. Thank you for inviting me. A little bit of background about myself. Like Helen said, I'm currently a postdoc at the University of Chicago in Paul Sereno's lab. My undergrad was in biology at Princeton, and then for my master's and PhD, I was in Bristol in the UK under Jakob Vinther, and I've been in Chicago ever since. And you can see what my favorite TV show is, judging by the art there. My research is pretty diverse. I do work on sexual selection in the fossil record, the evolution of feathers, of course, all sorts of things about dinosaurs because you have to as a paleontologist. But I would say kind of my main area is molecular taphonomy and what you probably are interested in hearing about today. And so these are all of the biological, geological, chemical processes that occur from life through death and fossilization all the way to discovery as a fossil. Anyway, so this is the most recent paper. It's sort of a culmination of work on looking for proteins and amino acids in various Mesozoic fossils. But some of you, I don't know who all is logged in, but some of you are probably co-authors on a lot of that work, and many of you are probably much more experienced in actual proteomics than I am. And so I kind of want to give a little bit of a broader view, sort of my motivation behind the studies, looking first at potential amino acids in bone apatite, followed by eggshell calcite. And then also kind of my perspective as a sort of biology, geology trained paleontologist about future approaches, in particular, what I informally call my attempts to make synthetic fossils, as well as searching for candidate biomolecules that might survive well in fossilization. Now, molecular paleontology was originally termed for studies that use sequence data from, ancient proteins or even ancient DNA, especially these cases with these ice age permafrost carcasses in order to, study them in a variety of ways, but in particular to make phylogenetic trees. However, more recently, the term sort of broadened out to include studies searching for any variety of soft tissue, so sort of non-biomineralized components within the fossil record. And these studies have proposed all sorts of very interesting things deep into the fossil record, including in Mesozoic dinosaurs. Vessels, blood vessels, blood cells, extracellular matrix, keratin protein, collagen proteins, even DNA. And there's a variety of methods that have been used. A lot of them tend to use these histochemical or immunological stainings, however. So the question is are we on the doorstep of Jurassic Park? Well, when I got to grad school, I began to sort of question some of these conclusions. And so I began my own work on this along with a very large team. So for the first study, I'm going to talk about dinosaur bone. Now, this is late Cretaceous Centrosaurus bone from Alberta, Canada. These are horned dinosaurs that died in what appears to be sort of a flooding event. So it's this massive bone bed where essentially if you dig down, you're almost guaranteed to hit bone, which made it easy to use relatively aseptic tools to quickly collect bone that had just been exposed or even was unexposed and surrounded by the mudstone matrix. I compared these samples to modern chicken bone gotten from the very exotic Tesco's grocery store in the UK, as well as Pleistocene age shark teeth from the beach in my hometown in Florida. And then I subjected these samples to a wide array of methods in order to triangulate conclusions, which I will go through. So if you demineralize these bones and examine them with lighter electron microscopy, what you see is that the demineralized dinosaur bone lacks these large organic massids that you see in the fresh modern bone or in the subfossil tooth. And not only that, but some of these vessel structures are infilled. And when you look at these under energy dispersive X-ray spectroscopy, you see that these dinosaur vessels are silicified. They are not organic. And now this is interesting because this infilling and silicification is something you could see with biofilms that are growing inside of the bone. Keep that in mind. If you look at these bones under IR spectroscopy, although you see some peaks in the dinosaurs that could potentially be affiliated with organic vibrations, they are, of course, less prominent and less clear than in the modern bone. And so clearly it's not organic rich in things like protein. If you examine these bones with pyrolysis GCMS, you see that in the dinosaur bone here in red, there are few complex organics compared to the modern bone, indicating degradation of organics. And in fact, the dinosaur bone most closely resembles the pyrolysis fingerprint, essentially, of the surrounding mudstone matrix. Not to mention the fact that sedimentary organics and soil organics, such as humic acids, can yield a variety of alkane-alkene products that were detected in the matrix. And so not a lot of evidence of intensive and high concentration organic material in these bones. Now, if we look at them with liquid chromatography, what we can see is that the dinosaur bones here in red are not only low in the concentration of their amino acids, but the composition of their amino acids is inconsistent with collagen. So what you see in the younger modern bone in blue or the subfossil bone, or excuse me, teeth in black, the composition is very different from the aseptically collected Centrosaurus bone. But with bone that was either eroded onto the surface or more heavily exposed during excavation, that Centrosaurus bone actually begins to converge upon an amino acid composition more similar to either the mudstone or even the topsoil, which is interesting, which is suggesting that recent amino acids are infiltrating these bones as they erode out and sit upon the surface. Likewise, you can look at the concentration through liquid chromatography of these amino acids. So the dinosaur bone in red has lower amino acid concentration than either the modern bone in blue, the topsoil, or the subfossil teeth. However, when these Centrosaurus dinosaur bones are eroded or heavily exposed, they do have higher concentrations of amino acid than in the various mudstone samples from this locality in brown or in tan. Now we can look at the racemization of these amino acids, and we see that in the dinosaur bones, they are not racemized, and they are heavily biased towards L isomers, suggesting that they are recent amino acids. Now, this is, of course, only in cases where the amino acids are in high enough concentration to even be detected. Some of these results are actually below the detection limit. And so what do all of these results suggest? Well, in totality, it does not give you a strong signature for endogenous collagen. But there are some things we need to continue looking at. This is not the whole story here. So for example, if you look more carefully at the pyrolysis GCMS data, you see that, yes, there isn't a clear fingerprint of, say, proteinaceous material or high organic content, but we do see organics such as kerogen, as evidenced in these alkane-alkene doublets. And this kerogen is, it's often described as a geopolymer. It forms over long periods of time through diagenesis. And so there, even though many of the amino acids are relatively recent, there is some relatively older organic material, perhaps not Mesozoic, but definitely not just completely modern. And so we can look at this further through radiocarbon accelerator mass spectrometry. And what we see is that the total organic content of the dinosaur bone is higher than the surrounding matrix. And so there's an enrichment of organic carbon in the bone. Now, the radiocarbon in that is not dead, so it's relatively recent, again, consistent with what we're seeing in the amino acids. However, the F14C is lower in the bone than in the topsoil or the mudstone. Now, this is interesting. And so based on this and using sort of a simple one box model, we estimate that about 26% of the carbon in the subterranean dinosaur bone, while it's buried, comes from the adjacent mudstone. And what this means is that it's not just a reflective of whatever is currently living in the surrounding mudstone. It's acting as a reservoir of sorts. There is some degree of residence within this bone. It might not be Mesozoic, mind you, but there's something going on and residing within this bone. Excuse me. And so we can look at this more deeply using fluorescence microscopy. So these are EDTA demineralized and then PI stained to stain for DNA that is non-nucleus bound. And so this is a way to look for essentially dead cells of microbes that are living within these samples. And what we see is that these blobs of supposed extracellular matrix, these long supposed dinosaur blood vessels, just light up with DNA. And we think that this is consistent. In fact, you can see some of these little spheres. We think this is consistent with a multi-cell aggregate forming organic vessels. So you have the appetite of the bone, and then you have microbes living within those pore spaces. So the original material is largely gone. And then what is actually forming is sort of a mold as a result of a biofilm to produce these vessel-like structures. In fact, if you attempt to measure the amount of DNA in these dinosaur bones using qubit fluorometry, there's an enrichment of 50 times more DNA in the dinosaur bone than the surrounding adjacent mudstone. Now, you can probably guess that this is not dinosaur DNA, but this is evidence of an active microbial community living within the bones. And of course, we can attempt to characterize this community. Here we see the results of a very simple 16S rRNA gene amplicon sequencing. And you can see there's a unique microbiome in the bone. So these 4 columns on the left are the bone. And then on the right, we have the mudstone, and then also the scraping of the outermost surface of the bone. And you can see that within the bone itself, there is a different composition of microbes than even the very immediate surface and very immediate mudstone surrounding it. And so this is very interesting, and it can explain some of these recent amino acids within the bone. And so the bone overall appears to be an open system. The original dinosaur proteins break down, they hydrolyze, and then those broken down fragments or free amino acids can leach out of the bone. But you also have the ability of microbes to infiltrate these highly porous vascularized bones to produce biofilms that then might mimic original structures such as blood vessels. And now there's all sorts of reasons why these microbes might preferentially choose to live within the bone. Phosphate, it's a great source of phosphate, being an appetite, which life needs. And then also these bones being so vascular, they're really good at wicking up moisture. And so there's probably a decent amount of water content in them compared to the surrounding matrix. And now we then more seriously in a subsequent study characterized this sort of dino microbiome, as I call it, using metagenomics. Surprise, surprise, no dinosaur DNA. But we did find 46 strains of novel microbes from 6 bacteria phyla and 1 archaeal phylum, which was very exciting, including some very unusual things. A large proportion of the microbiome was something called Eusebia, which I think was only really described from like Etruscan tombs in the the skin of sea cucumbers, which was a very odd microbe. And now, if you look at the metaproteomics of this bone and of this dino microbiome living within, so these are modern microbes living within the buried subterranean bone. Again, you don't see any dinosaur collagen. But what appears to be taking place is that the various taxonomic groups of microbes are performing different metabolic functions regarding, carbon, nitrogen, sulfur, iron metabolism. And this is a living community. They're active. They are live. In fact, at one point we saw a cell kind of dart across in one of our microscopy samples when we had demineralized the bone. But the growth appears to be very, very slow, which isn't really surprising if you're living inside a dinosaur bone buried underground. And this is interesting, of course, because it goes along with this really recent interest in the deep biosphere. These microbes, or even sometimes more complex things like nematodes, live in deep underground in South African mines or wherever they sample from. From my perspective, it's also interesting because it suggests that not only do microbes play a role in early stages of decay of a carcass, that the early rot carcass, perhaps either before or shortly after it's been buried, but that these microbes can continue living through the bone while it's buried underground during the middle and late stages of the history of this fossil. And so it's something to keep in mind that these bones are active habitats. And now, what about these original claims? Like I said, a lot of the original claims of DNA or proteins or whatever, they use these histochemical or immunological stains. The discovery of this active microbial community suggests that these could be, cross reactivity or false positives. The detection of kerogen, these sort of complex sedimentary geopolymers, could also be a source of cross reactivity, as well as any glues that are inadvertently applied to fossils as they're being excavated. And this also matches with some of the other critiques, such as the weak statistical support for some of these peptide sequences that have been purported from dinosaurs, as well as their potential source of contamination within the labs that have worked on these dinosaur specimens. Okay, so if bone is an open system and is maybe more interesting to a microbiologist as opposed to a molecular paleontologist, what about closed systems? Well, luckily, a lot of my colleagues have done a lot of the legwork here, and in fact, some of them might be listening to this talk right now. But biocalcite appears to be able to act as a very good closed system to trap original peptides and definitely original amino acids. And what's also very exciting is that methodologically, by applying a oxidative bleach treatment to biocalcites such as mollusk shell or bird eggshell, you can actually remove any proteinaceous material that is outside of those calcite crystals and could be contamination, and then examine only the intracrystalline fraction of the proteinaceous material within these samples. And so you can be more confident that you're not examining contamination. And these results have been really exciting for a variety of reasons. I mean, really, really solid evidence for endogenous original amino acids in mollusk opercula as far back as 30 million years ago by some of my colleagues, such as Kirsty Pinkman. And then I'm sure many of you are familiar with this work, some of these peptides found in fossil ratite eggshells. So not only are free, completely hydrolyzed amino acids capable of being trapped inside a biocalcite, but these short segments of some peptides can sometimes be found far deeper into the geologic record than you might have otherwise expected. And so struthiocalcin, for example, is protein that's involved in mineralization of the eggshell, and a particular region of it, a very short and acidic region, has been detected in higher latitudes, perhaps as far back as 9 million years old. But even at low latitudes, 3.8 million years old, which is quite surprising given the temperature regimes down there. And the hypothesis being that there's a high binding affinity for this particular region to the calcite based on the function of the protein, and that this binding stabilizes the protein, which can be thought of as essentially dropping the effective temperature around that sequence by about 30 degrees Kelvin, which would then slow hydrolysis. So it's sort of like a molecular equivalent to freezing this protein, and allows for the sequence to survive far longer than it might otherwise. And so essentially my thought, I had seen some of these papers on bird eggshell. I had been doing this work on the dinosaur bone. And I was thinking, okay, if avian eggshell, so birds being modern dinosaurs, if avian eggshell calcite can preserve ancient peptides, and if I take dinosaur eggshell calcite and I don't find any preserved peptides, then it's even less likely that their apatitic bones would, because as we've seen, the apatite is a very open system. It allows for the loss of original proteins and contamination of microbial communities from the environment. And so I began to look at eggshell with a whole team of people. I think there's like 20 co-authors on this paper. Originally took them up to York with Kirsty Pinkman and her lab. And now early on, I was looking at Argentine titanosaurs. So that was the dinosaur in the previous image, the long neck sauropod dinosaurs, which include the largest land animals to ever live. And these eggshells actually came from the commercial market, which was both a blessing and a curse. Obviously, they have less verification as to their source, but they're also free game for highly destructive exploratory analysis. And based off of the initial success, we then added to the samples later on, Argentina, Spain, China, not just titanosaurs, but also these duck-billed dinosaurs called hadrosaurs, using fossil samples from museum and university collections with better record keeping about their collection. Again, I try to use methodological triangulation whenever I can. And so just like in the previous study, using a variety of microscopy, spectroscopy, spectrometry on these samples. And when we look at these under light microscopy, you can see that although there is some recrystallization in some of these samples, overall, the morphology, the shape of the calcite structures within the eggshell, such as the surface ornamentation on the eggshell, which are very diagnostic, and as well as these columnar calcite crystals in cross section, are still preserved. Many of these eggshells have very dark cross sections, consistent with, say, high organic content. Now, we examined these under Raman spectroscopy. The Raman spectroscopy, it definitely shows two phases, the recrystallized and not recrystallized portion of the eggshells. There's various bands that are consistent with organics or organic polymers. However, there's currently a lot of debate about this method on some of these fossil samples and whether or not there are spectral artifacts as a result of background fluorescence in some of these samples. And so that's something to keep in mind. But we can use further methods to look into this. So again, pyrolysis GCMS between a dinosaur eggshell and a modern chicken eggshell. What we can see is that although there are pyrolysis products that you would expect from, say, amino acids in the dinosaur eggshell, you get a smaller set than you do in the modern eggshell. This could be, low concentrations of material in the dinosaur bone, or maybe less complex organic material. So instead of polypeptides, maybe more free amino acids. And we also see these alkane-alkene doublets, again, consistent with this sort of organic geopolymer kerogen preserving in the eggshells, consistent with the fact that these are very, very old Mesozoic dinosaur eggshells. Overall, the preservation is not as pristine as the modern eggshell. But what's really exciting is when you do the high-performance liquid chromatography to characterize the amino acids in some of these dinosaur eggshells. So if you look at, say, some of these titanosaurs in panel B or the Chinese hadrosaurids in panel E, you can see that there is a profile of amino acid composition that is very exciting. You have high glutamic acid, high glycine, alanine, and valine, possibly valine. And those are really the only amino acids that consistently come out across all of our replicates. They are depleted in aspartic acid and serine. And this is the exact pattern that you would expect from subfossil avian eggshell, as well as avian eggshell that's been heated either naturally or experimentally. And so when these results first got printed off of the chromatograph, Kirsty saw them and started shouting and jumping up and down. It was very exciting because this is exactly the sort of profile you would expect from a very, very ancient eggshell, essentially kind of at the very end of that diagenetic continuum of degradation of eggshell proteins. Now, there is variability between the different localities. So you can see some of these titanosaurs do not give as clean of a result. And this appears to correlate with the amount of amino acids overall within the sample. So the more amino acids that are preserved in the dinosaur eggshell, the better the profile is to that which would be expected from an ancient eggshell, which I suppose is not too unexpected. Now, again, we can look at the racemization patterns in these amino acids, and unlike the dinosaur bone, these eggshells are consistently fully racemized, suggesting that they are very, very ancient. Not only that, but we see conversion. We interpret conversion of serine to alanine, which is also a diagenetic process within these amino acids in these eggshells. And then finally, the samples of eggshells that were bleached, we were talking about that oxidative bleaching method that removes contamination. When we compared the bleached eggshells to those that were not bleached, you get very similar results. And this would be consistent with the idea that most of the amino acids we are detecting are indeed the intracrystalline amino acids. So whatever is left behind is the stuff that is presumably locked away within this closed system within the calcite crystal. Now, of course, you find all these ancient amino acids. The question is, well, is there peptide preservation? And one of the first ways you can go about looking at that is, well, can we sequence any peptides? So when you use liquid chromatography, tandem mass spectrometry, the detected peptides, the only ones that are well supported statistically are those that could potentially come from laboratory contaminants. So you get a lot of human type keratin, for example. And so as hard as we look, and we replicated this in 2 different laboratories, you don't find any dinosaur proteins. So you find really ancient amino acids that appear to be consistent with original eggshell amino acid, but you don't find any proteins. And so this suggests that whatever original proteins were in the eggshells, they are now fully hydrolyzed. And those amino acids are either fully or largely hydrolyzed and not within their peptide bonds and within a chain. Now, we also used time of flight secondary ion mass spectrometry on these eggshells. Unfortunately, the data wasn't amazing. Calcite of the eggshell and the embedding epoxy of the thin section sort of dominate the signal. We did get some secondary ions that are potentially consistent with amino acids and other organics, but not much we can say with that data other than it's largely consistent. Okay, so in conclusion, we have a variety of eggshells that we looked at of different ages, different localities, each one of these columns. Some of them have weaker evidence of amino acids. Some of them have very strong evidence of amino acids. But overall, we have good evidence for original endogenous amino acids in dinosaur eggshell calcite, possibly as old as 86 million years ago, which is very exciting. Another aspect that's really interesting is this kerogen result, these organic geopolymers that we're finding. They could, for example, derive from the original lipids. So these are like these Maillard type reactions, or similar to Maillard-like reactions, I should say. Cross-linking, polymerization, and perhaps the formation of these geopolymers help to close the system within the calcite. And so they might actually be playing a role in the preservation and trapping of these amino acids. And now, with only about 3, maybe 4 amino acids detected in these eggshells, we clearly don't have genetic data. This would be the equivalent of removing all but 5 letters, I guess, in the English alphabet from a book. You're not going to have sentences. You're not going to have paragraphs. You're not going to be able to make sense of it all. What's funny is I made these images with AI, and it actually got the hands and fingers correct, but all the words are complete gibberish, which I thought was kind of funny. Okay, so if we don't have genetic sequences, well, what can we say with this? Well, we're limited, but there's still a lot of exciting potential for this, uh, fossil biocalcite research. So even with a relatively restricted set of amino acids, their relative concentrations, that profile, that compositional profile, still might be reflective of higher taxonomy. So for example, eggshell versus mollusk shell. You also have the potential of really interesting stable organic isotope studies where if you know that, say, the amino acids within these calcite crystals are likely to be endogenous and not representative of contamination, then any stable isotope work you do on these samples, you can more confidently ascribe to the specimen or the organism itself rather than some sort of environmental contamination. And then, of course, calcite and calcification is very broadly distributed among animals. And so it allows you to potentially study a wider range of things very deep into the fossil record, potentially. So one of the things we want to look at are some of these Paleozoic trilobites, in particular, their calcitic eye lenses. Okay, so I hope I'm doing okay on time. I'll try to go through the rest of this relatively quickly, but I want to talk about sort of the methodology. So I want to talk about diagenesis. Now, this is the aspect of taphonomy from life to discovery that I focus on quite a bit. Now, many people, you have different definitions for diagenesis. I tend to view it more as it's sort of like how you think of the oil and gas window. This is the heat and pressure during long-term burial from geothermal heat from overlying sediments that can result in volatilization of organics and polymerization of organics. And in terms of looking for ancient fossil biomolecules, I often consider this as essentially the ultimate hurdle, barring any sort of late stage metamorphism, for example, or extensive weathering of the fossil once it's exposed. You have to be able to survive diagenetic maturation if you want to preserve as a fossil biomolecule. And so I focus a lot of my research on this aspect as a result, sort of like the final boss of a video game. And, I'm far from the first or only person to try to study diagenesis experimentally. Experimental or artificial thermal maturation has been a staple in the study of sedimentary organics, particularly fossil fuel research, and for many years and decades. And the basic idea behind experimental maturation is kinetics. For many chemical reactions, you increase the temperature, you increase the rate at which that reaction happens. But of course, you also have to be careful about not exceeding activation energies for chemical reactions that would otherwise not occur during diagenesis, such as when it's in this oil window phase, or even higher in a lot of these fossils. And so you're using heat to try to simulate long-term diagenesis, but on laboratory timescales without exceeding unrealistic activation energies. And so I don't know about you, I don't have millions of years to run an experiment, but 24 hours a week, a month, that's far more reasonable. And so it's sort of analogous to pressure cooking, when we have these Instapots here that can cook a turkey in 20 or 30 minutes, whereas us Americans on Thanksgiving would otherwise be putting it in an oven for about 3 hours. And so using heat and pressure, you can simulate degradation on shorter time frames. And so we run our experiments hot. The temperatures we use in these experiments are higher than what these fossils would otherwise experience, but that's to account for time. And the reason that we increase pressure during this cook is so that you maintain aqueous chemistry. You keep water in the liquid phase, so that you continue to do the proper aqueous chemistry, as opposed to desiccating out the sample. Also keep in mind that the vast majority of fossils are deposited in sediments that were brought in by the action of water. So most fossils are sort of waterlogged for much of their taphonomic history. And so in our molecular fossil hunt, we can look directly at fossils. We can look actually at the geologic record. And generally speaking, as you get older, older fossils, older sediments, there is a correlation with the depth that that fossil would have been buried at and the geothermal heat and pressure. Obviously, this is not a perfect correlation, but typically speaking, that's how it is. And then what you can do is you can mirror this. You can parallel this experimentally by applying treatments such as heat and pressure to various samples to sort of mimic this spectrum, this continuum of degradation. The idea then being that in both your sort of fossil record and your experimental conditions, you get a series of relative stabilities of different biomolecules, A, B, and C in this case. And then, of course, you can use the fossil record to calibrate your experiments, the treatment, how much heat, how much pressure, in order to get a result that matches with your sort of fossil of interest. And so there's all sorts of interesting avenues you can go with thermal maturation, some of which are less expected. So this was sort of a surprise result. If you take turkey skin and you moderately mature it such that it doesn't completely break down but is largely degraded, and then you look at that under the low pressure conditions of a scanning electron microscope, I observed these folds and pocks and blebs, which were very similar to what had just been published at the time of putative dinosaur red blood cells, which also appear to be just folds within a continuous organic material, pot marks, blebs. And so this sort of surprise result through maturation suggests that these are not red blood cells, but maybe degraded biofilms from microbial communities living within the bone, or potentially degraded consolidants from glues that might have been applied to the fossils. And so in this case, those structures are not biologically formed blood cells. They are self-organizing structures that have a wider range of sizes and shapes than what would otherwise be produced through controlled biological processes. And then other claims of dinosaur blood cells might also match this, but rather than organic folds, they might be things like iron oxide or pyrite framboids. So for example, here's two putative dinosaur blood cells next to each other, and you can see that there is a wide range of size, even in completely adjacent structures. But what I'm really interested in is using maturation as a form of independent validation for conclusions you make when studying the fossils. And the work I do here, I sort of informally call synthetic fossilization, where I take a sample, I bury it in the sediment, compact it with a hydraulic press, and then use a heated coil in a maturation chamber that is pressurized with gas. Gas could be air, gas could be inert nitrogen, to then undergo the actual thermal maturation under high heat and high atmospheric pressure. And so what you're doing is you're taking a known tissue with a known composition, and then you're exposing it through simulated diagenesis to hopefully infer what components are thermally stable and which are not. And the sediment pore, the compaction within the sediment that we do, acts as a filter. And so it allows you to trap the thermally stable components and then allows for the escape and the leaching away of unstable components, kind of like what we were talking about with bone, where broken down proteins can leach away. And we have a lot of success with some of these experiments. So you can take bits of lizards and feathers, and you can expose it to sediment encased maturation. You get brown bones, you get these brown stains around the bones, you get loss of volume, loss of proteinaceous tissue like muscle, loss of fats, loss of connective tissues, collagenous connective tissue. And they resemble, in a lot of ways, genuine carbonaceous compression fossils from some of these exceptional sites of preservation, some of these Lagerstätten sites. And then even at the microscopic level, they're similar. So for example, these pigmented structures in the skin, when viewed under electron microscopy, you can see the melanin pigment is still preserved within their melanosome vesicles. So each one of these little rods or spheres, or these melanosome vesicles, just as you see in these carbonaceous fossils, where the unstable components of the proteins and the fats have been lost and leach away out into the sediment, but the stable components, such as the melanin pigments, survive. This was my PhD advisor's big discovery. Jakob Vinther was the preservation of this melanin in fossils. We also have success with other tissue types that are known to survive fairly well organically in the fossil record. So we got a leaf here, we got the cuticle of an insect here. They survive very well through sediment encased thermal maturation, consistent with the fact that we find organic fossils of insects and plants in many different environments. Now we can look at this more closely down to the chemical level. So this is work based using time of flight secondary ion mass spectrometry, led by Arindam Roy. And what we found is that when you synthetically fossilize, quote unquote, these feathers, not only do they look like genuine fossil feathers, but we appear to be able to simulate a lot of the diagenetic sort of continuum here, where you lose volatile small components and you increase your cross-linking polymerization of this melanin pigment. And so most of the variation, principal component one, is this diagenetic continuum of temperature, and also whether you do it in sediment, which is an open system, or a closed system capsule. And the only real difference between these experimental samples and the fossils, which are up above, is in their lipid content, which might be due to the fact that we did not expose these samples to decay or weathering. And so the lipid loss we see in fossils might be due to some of these other processes that we could in the future experimentally simulate, but we did not there. Some of the work I'm doing right now involves tree resin. So I've taken resin and exposed it to sediment-encased thermal maturation, and you get stuff that looks a hell of a lot like amber and copal. You get these conchoidal fractures, you get this brown or amber color. It's semi transparent. You get some of these rings, like these flow rings within these structures. And so I'm interested in the future to study this amber experimentally, to see just how open or closed the system of amber is for anything that's included within it. And again, you can see that the sediment is providing a useful role here. So this is a big blob of tree resin that was matured, and you can see that there's this front of volatile material that is leaching out and away from the hardened body of the resin. And so we're simulating a more naturalistic environment than would otherwise take place if you were to do thermal maturation within a completely sealed capsule. And so with all of this information, we can come up with sort of a guide to looking for molecular fossils based off of their sort of thermal stability. And so a useful starting point is thinking, and this is something that Jakob very much impressed upon me, is starting off with your organism and considering what in the organism as it is when it was alive is already stable through fossilization. And of course, this explains the vast majority of the variation in preservation potential, because biomineralized tissues survive best in the fossil record: bone, teeth, shell. Not really surprising that mineral can survive in the rock record, after all. But what about organic material? Well, some of the things I would look for are thermally stable molecules that can survive diagenetic maturation. So you're looking with things with few reactive groups. You don't want a lot of nitrogenous proteinaceous material. Aromatic rings help through some of their resonance behavior. Cross-linking and polymerization can create a very insoluble compound. And then, of course, low aqueous solubility. If you're in waterlogged sediments and you are soluble in water, chances are you're more likely to leach away from the sample. So, we've been talking about kerogen, this sort of geopolymer, which is a prime example of that. But you can also go back to our oil window, thinking about diagenesis, catagenesis, metamorphism. And you see this pattern from more complex organic materials. And as they're more matured, they become much simpler. Obviously, graphite and diamond being the most stable. What this allows you to do is you can take your various classes of biomacromolecules and roughly rank them in order of their fossilization potential. Obviously, nucleic acids, DNA, RNA are the lowest. We have permafrost from permafrost DNA, environmental DNA from about 2 million years ago, which is actually a lot longer than a lot of people had originally thought. But this is nothing compared to some of these lipids and pigments, which are from bacteria measured in billions of years. So, for example, these hopanes from bacteria, which are sort of their equivalent to steroids from, 2.7 billion years ago. I'm not sure. There might have been actually more recent work which has pushed that back. Uh, but not only do you want to think about the biomolecule itself, you also want to think about the environment in which the fossil or the molecule is preserved in, right? The surrounding matrix, whether you're talking about the matrix of the tissue or the matrix of the surrounding sediment. Obviously, you want closed or limited systems. You want to trap your stable amino acids, for example, in biocalcite, but you want to prevent microbes from infiltrating your bone, your fossil. And so you want low flux in or out of the system. Again, biocalcite's a great one. But just generally speaking, sediments that are maybe finer grain from low oxygen environments, and then of course thermally immature sediments are going to be prime candidates for good organic molecular fossils. And overall, this allows you to sort of come up with a framework for how you do studies on these ancient putative fossils. And I'm really trying to kind of aspire to this sort of standard where you have an analytical side where you're running a fossil through mass spectrometry, for example, but you're also comparing it to modern analogs, so modern tissues. If you have a fossil feather, for example, you want to compare it to a modern feather. If you have the fossil, you want to have controls. So, for example, this could be the sediment. If you suspect that it's been exposed to glue during excavation or preparation, you might have glues as a control. But then not only are you examining these various modern fossils and controls, you also want to parallel this with experimentation, whether decay, maturation, whatever is relevant for your question at hand. And so, for example, in this study, I have modern and fossil feathers, I have the sediment control, and then I also have a thermally matured feather material that you can compare to. And so with this in mind, I will end on a very brief discussion of one of my ongoing projects in which I saw that, there's a lot of studies on cholesterol having great fossilization potential, particularly its cholestane version, so its fully diagenetically altered version, surviving deep into the fossil record. And Precambrian deep, and then realizing that these sex hormones are biosynthesized from cholesterol, and that they have different numbers of carbons in their underlying skeleton, such that even if they are fully altered into their sterine versions, they should still be distinguishable from each other as well as from other classes of steroid. And so again, I'm trying to follow my ideal here, looking at fossils, looking at experiments. We have estrogen, just standard purified extract of estrogen that we expose to thermal maturation. In this case, this is the sort of traditional version in which it's in a sealed capsule. But what you can see is that even under extremely harsh conditions, 245 Celsius, 250 bar for a day, this molecule is essentially unaltered. So it is extremely thermally stable. If you look at modern bird bones that are not, necessarily fresh, they're relatively dry bird bones. They're not dripping in blood. This is not the blood itself. This is not the gonad material. This is just the bone. You can easily detect estrogen if you mature them either within sealed capsules or within my sediment encased maturation protocol. You can continue to detect these estrogens. You do not detect the estrogen in the surrounding clay within that experimental synthetic fossil. And so all of this is very consistent with, estrogen or estradiol to be specific, having high fossilization potential and potentially useful in sexing fossils. Now we examined a variety of kind of a hodgepodge of Mesozoic and Cenozoic fossils. A lot of them, some of them, we have potential trace amounts of some key ions that would be consistent with estradiol, eluting at the proper time. But I'll fully admit they are not, 3x above the baseline. So they're extremely weak, even if they are genuine. This particular specimen of a duck-billed dinosaur actually gave such a good signature of estrogen that we're not even sure whether this is a positive. It might actually be contamination, but some potential positives and interesting results from the fossils, but we haven't really looked at anything from an environment that's actually known for organic preservation. It's just sort of a random collection of fossils I was able to get my hands on. So we're continuing to look and continuing to pursue this project. Anyways, I'll end with that. I have no idea how much time I just took up, but hopefully not too long.
Thanks,
So
Evan.
yeah,
We've got
a lot of people to thank and acknowledge. Like I said, I don't know who's logged into the talks. Your name might be up here otherwise, but there you go. So thanks for inviting me.
great, thank you. Yeah, we've got like maybe 5 or so minutes for questions, or a little bit longer if we need. So if anybody has any questions, they can either pop them in the chat or they can put their hand up and then they can just speak and chat to Evan directly. I have a question straight away. I was wondering, with the amino acids you have in the dinosaur eggshell, what kind of concentrations are you getting with the amino acids that you think are endogenous?
They're pretty low. As for a number offhand, I would have to double check. That is actually something that I think should be done in future work is a much broader sample comparing concentrations to, let's see, is this, sorry, I'm trying to find underneath. No, that's just the, okay, whoop.
But was it more in like the sort of like hundreds or thousands or
Well, here,
millions?
I would say this panel right here.
Mm-hmm.
So this is total hydrolyzable amino acids
Yeah.
in picomoles per milligram.
Okay. So
One of
somewhere
the
roughly
samples
high.
gave a really high result.
Yeah.
But for example, so gray and blue, light blue and dark blue, those are the dinosaurs that did not give very good results. So the blue, those are these blue, and the gray are the dashed lines here. Yes, the dashed lines are the gray. So what we see is that once you get sort of above 500 or 1,000, then you are able to detect a pretty decent profile, which matches what you would expect from a diagenetically altered bird eggshell.
And so how confident can you actually be that those are endogenous and are not part of the contaminants that you can see with the LC aspect?
Well, I mean, that's always the question is when you're dealing with low stuff. But there's just various lines of evidence. One being, if it's contamination, then you have to explain why the profile matches that of a diagenetically altered modern dinosaur eggshell. So, for example, why is it that you consistently get this strong glutamic acid peak followed by your glycine and alanine and then maybe valine, as opposed to the reverse, where glutamic acid is really low and you have really high, you see what I'm saying? So the profile itself is consistent. The fact that you see it with the higher concentrations, the fact that they're fully racemized suggests that they're ancient. The fact that you have other organic compounds that are consistent with very ancient organic material, this kerogenous organic geopolymer. The fact that you can use this bleach treatment to better isolate the intracrystalline amino acids. All of the groundwork that's been done by, Pinkman, DeMarchi et al., all those people that have really put the work in and shown that these eggshells, the intracrystalline region, can act as a closed system. And so, yeah, it is very difficult because you're dealing with low concentrations, but that's why I use many different methodological approaches. And if they're all consistent, then you can be a little bit more confident. What else? Serine to alanine, that's another indication that you're dealing with ancient material. The fact that you get similar results between bleached and unbleached could be potentially consistent with the fact that most of the amino acids are within those crystals. The paper that we published has like a probably too long section that I wrote going into every minute detail about this. And so I would recommend looking at that if you want to be bored to death. But yeah, I mean, there's always uncertainty with it. Could these be? Sure, but what I can tell you is they're definitely more promising than the results we got with the dinosaur bone, right? The
Great.
appetite.
Yeah.
That's for sure. Um,
We have another question from Lindsay in the chat. So she said, thanks for a great talk. You mentioned an interest in the deep time soil microbiome when you were assessing dinosaur bones. Can you talk more to the potential of fossils as reservoirs of soil microbiomes through time?
talk more. I would like to say yes, but this is where I lean heavily upon my microbiology collaborators. Um, we are in the process. Unfortunately, I'm a little low on funding, but we are in the process of doing this study again on a different locality, a different fossil locality. So these bones come from your very typical fossil sites, particularly your very standard dinosaur dig sites. This was in Alberta, Canada, relatively low vegetation grassland. Gets hot in the summer, cold in the winter. We have a site from Missouri in the Ozarks. I mean, this is deep backwoods Missouri. And it's a clay pit, and this thing just floods all the time. I mean, this water or these clays soak up so much water. It is the weirdest site I've ever dug dinosaurs with. And we've sampled some of the fossil turtle bones from there and sort of slowly getting those samples prepared for metagenomics. And essentially, what we're going to see or we're going to look for is, do you get a lot of similarities to this fossil microbiome? Or is it wildly different because the environment is different? In other words, what's more important, the fact that you're a microbial community living inside of a fossil bone underground? Or is the surrounding environment more important as to what you're actually doing? And I imagine there's going to be some similarities. I mean, you're living in a bone that is a rich source of phosphate either way. And so there's going to be similar metabolic pathways in that sense. But it'll be interesting to see just how much variation there is in these microbial communities that are living within fossil bone while they're buried underground.
Have you
Um,
have you done with the metagenomics in the DNA? Have you looked at if any of those, if that's any of that's ancient DNA to see if the types of microbes that are living in the bone has changed over time? So, or are those all modern?
so I'm trying to remember the paper. um, our interpretations, at the very least, were that this was we interpreted these as modern. We did not go that far into looking for characteristics of ancient DNA within these bones, but the radiocarbon results suggest that there is some sort of residence time within the bone as a system. These microbes they infiltrate the bone from the environment, but then they clearly proliferate and they hang out and they live in there. And so, I didn't get into this in the talk, but some of that kerogen that we found in the bone, could that be dinosaur kerogen? We jokingly called it dinosaur oil. It's not actually, but could that be derived from, say, the lipids of the cell membranes within these dinosaur blood vessels? Maybe. But if these microbes are living inside the buried bones for long periods of time, then some of that kerogen could actually just be ancient biofilm, essentially, like ancient microbial organics. And so we haven't really looked at ancient DNA, but it could be that some of the ancient, or excuse me, it could be that some of the kerogenous material and some of this older carbon that we're detecting could be not necessarily dinosaur carbon, but it could be carbon from sort of previous iterations of these microbial communities. But that would be interesting to look at. And no, we haven't looked at it. that's definitely out of my wheelhouse. So I'd be
Yeah, it would be cool though, like to see if it changes over time. Yeah, it's nice. Lindsay says thanks so much. Do you have any other questions? You can either pop them in the chat or you can unmute yourself. But we're 5 minutes over, so if anybody has any, they can pipe up now. Alison, do you want to unmute yourself?
Hello. Hello. Thanks,
Hi.
Helen, Lindsay, and the whole PAASTA team for the opportunity. And also thanks, Ivan, for the amazing presentation dissecting the ancient molecules. I'm Brazilian-based professor in translational medicine here at Federal University of Ceará. And nice to meet you all. So for human remains, we have it's relatively easy to choose the petrous bone and the tooth as to proceed with the extraction. But how can we choose the best part of the dinosaur bone or other long bones? And is there any point of measurements that is the best to try some Raman spectroscopy or some ATR? And also about the age of the organism contributes to good retrieving these molecules or not. So it's the part of the bone and the age of the organism. We have some standard to follow.
Okay, so if I think I'm following all of your questions. So obviously the age is going to be a factor, and what really needs to be done is a broad survey study where you're taking samples of a wide variety of ages and you're studying the degradation across things from thousands of years ago, tens of thousands, hundreds of thousands, millions, tens of millions, hundreds of millions of years ago. I've been focusing on Mesozoic specimens, particularly these Cretaceous dinosaurs. So the Centrosaurus that I studied here, this is a late Cretaceous dinosaur. I think it's somewhere along 75, 76 million years old. The dinosaur eggshells got up to 86 million years old. But the reason why I jumped into the deep end of the pool, so to speak, is simply because previous work had suggested very exceptional preservation. And so I was essentially, revisiting or reconsidering that work. But yeah, you're right. I mean, you should sort of start at the shallow end of the pool, the younger material, and work your way back. And this, I think, goes sort of hand in hand with your question about which type of bone we sample. So first of all, you don't always have a choice as to what bone is preserved. You kind of get what you get sometimes in terms of what is actually found and what is still missing on a particular specimen that might not be complete. Unfortunately, dinosaurs did not bury their dead in a nice little spot, that you can have, everything all nicely preserved. And so you're sort of limited on that. My guess is that, yeah, like dental enamel, the petrous bone, those are going to be better sites of preservation deeper into the past. But we're dealing with orders of magnitude when you're jumping from, say, archaeological samples to dinosaurs. And so without looking at equivalent tissues in dinosaurs, now we did, actually, we should probably get this published at some point. We did look at dinosaur teeth, a few dinosaur teeth in York, doing amino acid racemization, amino acid analysis using liquid chromatography. And those came up with just like the bone. They did not give any indication that there were original amino acids. Now that I think about it, we really should publish that because this is a question I've gotten several times now. And so without really basing it off of much of anything, if I had to guess, my guess is that if you're talking about appetite, it doesn't really matter once you're hundreds of millions or tens of millions of years old. It's only going to get you so far. But for archaeological remains, yeah, sure, that's definitely where I would look first. But yeah, we're just talking orders of magnitude different ages. Not to mention the fact that you know, the northern polar ice cap is what, only 5 million years old. The southern polar ice cap is only 30 million years old. And so any permafrost associated with those ice caps are going to be restricted in age. The climate gradient of the entire Earth was much shallower back in the Mesozoic, for example. So the higher latitudes were relatively much warmer. The overall temperature of the Earth was much warmer. So not only are you dealing with time, the environments in which they experienced their first, 50 million years or so, 50 million years or so of taphonomic history would have been much warmer, no polar ice caps. And then, of course, you're talking about any additional geothermal heat. If they get buried underground, even in relatively thermally immature sediments, geologically speaking. And then I think you asked something about Raman or IR. I mean, I try to use Raman and IR whenever I can. I try to use as many methods as possible. There is currently debate, particularly with Raman spectroscopy, although I imagine it holds true in IR as well, about avoiding spectral artifacts. So that's sort of a methodological question that's been debated sort of currently in the literature. I'm not sure if that answers your question about it, though.
Great, thank you. Lindsay said she had to leave, but thank you very much for a great talk. If there are any final questions, please put your hand up and unmute yourself, or you can pop them in the chat. Otherwise, I think that we can say thank you very much to Evan. That was a really, really great talk, and covered lots of bases, which are all super cool, super interesting. It's unfortunate that Jurassic Park's not going to be happening anytime soon by the looks of it, but anyway, it's good to know these things.
I tried to end on a more optimistic note, which is if you consider what your prime candidates might be, there is potential for all sorts of exciting molecular paleontology work in the future, even if it doesn't have to be DNA or proteins. For example, my advisor, Jakob Vinther, invented an entire field of study reconstructing the original melanin-based coloration of feathered dinosaurs. It was cool enough that I moved across an entire ocean onto a different continent to study with him specifically. I'm ashamed to say I did not know where Bristol was on a map when I actually moved into my flat there. But that work was so cool to me that I was willing to head all the way out to the UK to do stuff like that. So no, there's a lot of exciting stuff if you just kind of think about what is most likely to preserve.
Great. Well, thank you very much for an amazing talk. And yeah, there's Megan said thanks, Evan. Nice to hear everything all together. That's great. So thank you everyone for coming. And the talk will be put up on the PAASTA YouTube channel as well. Thanks.